Hello everyone, welcome at the BiCIKL session. BiCIKL stands for Biodiversity Community Integrated Knowledge Library. This is actually a 3 years EU-funded project, but for those 3 years it became well known, especially the TEDweek audience, because this is our third symposium at the TEDweek. And for the first part of the session, I'm giving the floor to my co-organizer, also partner in BiCIKL, Wouter Addink from Naturalis. Wouter, please.
Thank you, Lyubomir. Yeah, this is session SYM09. This is about biodiversity knowledge linking progress after BiCIKL and beyond. We will have 4 talks in this first part of the session and 5 talks in the next part after the coffee break. And that means that we have also some time left after these presentations to do some further discussion. I would thank the sponsors of the conference, and especially the Diamond sponsors, so Max Communications, Delta Designs, and Pictory. I would like to remind everybody that we will look at any questions that are raised by the virtual participants of the sessions, and so we will answer them here if there's time. And I would like to remind the speakers that we need to keep time because there's no time after the presentations in the first part of the session. So you only have a maximum of 15 minutes. Try to keep it to 13 so that we have some time for 1 or 2 questions. I would like now to give the floor to the first speaker, who is Lyubomir. Please go ahead.
Hello again, I'm Lyubomir Penev from Pensoft, and I'm the coordinator, or was the coordinator of BiCIKL. So, BiCIKL, as I mentioned, is a 3 years project with 14 partners from 10 European countries. The total budget was nearly 5 million euro, and it ended just last April. Those 14 partners were represented in BiCIKL by 16 research infrastructures, and you can see on this slide some very well-known brands of world level and well-recognized research infrastructures and services. And they represented actually the different data types they handle. For example, some handle taxon names, some specimen records, some manage sequences, and so on. So the main results of BiCIKL are mainly in the field of 2 main topics. The first one is improved access to fair data between research infrastructures and at each separate infrastructure, for which we developed several tools and services. The second one is building links, especially bidirectional links between research infrastructures, and also along the line specimens, sequences, species or taxon names, analytics, publications, which finally should end into data which will help building the biodiversity knowledge graph. We intended and did it to build a new community of users. Users we understand as research infrastructures which help each other in their services and data provisions, but also between research infrastructures and researchers and users. BiCIKL was really focused on the biodiversity literature. It created several workflows for automated extraction of data from the literature, and also new semantic publishing workflows, which actually resulted in a common data pool coming from 2 sources. The first one is the historical literature, from where the data have been extracted by Platzi from the PDFs, and the second one by newly published literature published in XML, where the data go automatically into the same data pool using the same identifiers and also the same ontologies and so on. Also, we developed several practices and standards for use of persistent identifiers in the different infrastructures participating in the project. And also community engagement was ensured by developing workbenches and curation methods for data at the different research infrastructures. The key product of BiCIKL is the Biodiversity Knowledge Hub, which is actually a web portal. And on that web portal, which is different from the project website, we summarized all main results in terms of guidelines, protocols, services, and one can find what we have done in BiCIKL on this web portal. The central point of the Biodiversity Knowledge Hub is the so-called FAIR data place. FAIR data place is just a catalog of services. And on that catalog, we list 16 services, 7 of which have been developed in BiCIKL, and the others have been significantly improved. And all those services are maintained not by the hub or by the project, they are maintained by the research infrastructure who developed those services, and they are fully responsible for their maintenance. Just example of some of those services. For example, the Biodiversity PMC puts together the biomedical literature from PubMed Central, that is PMC, with biodiversity literature. But Patrick Roche, who is here, will tell us more about that. Another one is federated semantic search for research data built by LifeWatch. The service is called LifeBlock. In BiCIKL, we built actually we developed the protocols and build infrastructure for assigning DOIs to specimens in collections, and that service is already working. It will be demonstrated today as well. Platzi built access point to a really rich pool of data they extract from the literature through treatment bank. Also, nano-publications for biodiversity is a new service and new workflow, which was built by Pensoft and Knowledge Pixels during BiCIKL. And there are some more, but I don't have the time to present all of them. There are leaflets and flyers just on the table in front of the door, and also at our booth in the hall below. So also the end users, researchers have been engaged in BiCIKL by open call projects. We had actually 3 pilot and 8 open call projects, which passed first evaluation procedure. And at the end, they aimed, and they did that. They tested the services and data access tools we have developed in, let's say, real-time research work. The research team were from several parts of the world, not just EU. BiCIKL, just in numbers, we have built 2 websites, 86 publications, of them 37 articles and many conference presentations and so on. 2 journal article collections. All those publications can be found in the project branded collection of articles in Rio Journal. 4 TEDx symposium, including 2 sessions of one and the same symposium during the present TEDx. 29 best practices and 65 recommendations, which are published on the Biodiversity Knowledge Hub website, and many thousands of users. We also submitted and published 2 policy briefs to the European Commission, one of them stressing upon the importance of further developing of mechanisms to link fair open data. And the second one about the importance of mobilizing the data which is closed or imprisoned, as we say, in the PDFs of the legacy literature. So what next? After BiCIKL, we submitted BiCIKL Plus. Unfortunately, it was not funded, but it got really high score, almost at the highest possible score. We'll plan to resubmit it again, but I want to tell you what we plan to do actually there. The next step will be to build the so-called biodiversity supergraph. This is a kind of sexy title to explain our idea. This is a centrally orchestrated and AI-assisted system of tools and services which will manage the different triple stores provided by the research infrastructures which participate in that process. So the research infrastructures are supposed to be able to export their quality checked data in RDF, and then a central system will make federated search and federated exploration of those triple stores. That's the basic concept behind the biodiversity supergraph, and it's built on the condition that we should use artificial intelligence tools and LLMs only on the basis of really adequately curated, semantically structured, and interlinked biodiversity data. Otherwise, we all know about the problems which might arise by using AI without control on the data they use for training. This example on the domain of literature, you see above that there are main data types handled by the different research infrastructures like specimens, genes, taxa, and so on. And the biodiversity literature domain is presented here with several different services which handle the data in different ways. Some extract the data, some convert the data into RDF, some provide really a powerful search mechanism inside the literature, and so on. Some publish nano-publications, as you see there. And in the center of this ecosystem of biodiversity literature services are actually the biodiversity literature repository based at Zenodo at CERN. This is just a repository of extracted and semantically annotated data like taxonomic treatments, whole papers, images and figures in PDFs, tables, and so on. And the second central point is the Biodiversity PubMed Central, which again will be presented, let's say, in this session. And because we all know that very large triple stores have some problems with performance, at least at the current stage of development of the hardware and software, therefore we launched the concept of supergraphs on demand, which means a subset of linked open data which will be created on purpose to serve specific research questions or use cases, which we hope will significantly enhance the scalability and use and reuse of biodiversity data, which are put together in this advanced way. And this is from me. I really want to thank all our partners, researchers who participated in the project, users, reviewers, for the great privilege to coordinate this exciting project. Thank you.
Thank you, Lyubo. Nice in time. So we have time for one or two questions. I didn't see any questions in the Zoom meeting. So let's go to the audience in the room. Are there any questions for Lyubo? No questions? Okay, then we move on to the next presentation by Teodor. Go ahead, Teodor.
Thank you, Wouter. So, hello everyone. I'm Teodor, and today I will talk about nano-publications as a universal method for publishing, annotating, and sharing biodiversity data. Before I start, I would like to see how many of you are actually familiar with the concept of nano-publications. Okay, so good. for those of you which are not, I will explain briefly. So the nanopublications is a very simple bit of information or the smallest bit of information which can be published. It consists of assertion, basically a statement, provenance, which says who made or on what basis this assertion was made, and the publication information, which tells us who actually created this nanopublication. During the BiCIKL project, together with Knowledge Pixel, which is a Swiss company, we have created a couple of different pilots for using this technology for sharing, publishing, and annotating biodiversity data. So we have integrated this in our standard workflows for two of our journals, Biodiversity Data Journal and Research Idea and Outcomes Journal. So the nanopublications, they are, as I said, universal. You can create different templates. For example, Specimen A is identified as taxon concept B. Taxon A interacts with taxon B. Taxon A inhabits habitat B. Research publication cites specimen, or research publication cites gene sequence. Basically, you can create different templates, depends on your needs or the information you would like to track. So how you can publish a nanopublication starting from a journal? You go on the journal website, there is a button, Publish Nanopublication. So you go from there. In the Nano Dash, this is the platform developed by Knowledge Pixels. You can select one of the templates, which is the predefined templates, which are very specific for the biodiversity domain. So you can fill the data in those templates, and once you did this, they will be published and visible on the journal website. So when you publish these nanopublications, you can put their links in your research publication, and this is the way how this is visualized in a research article. So basically they are as a separate section at the end of the paper. There is a human readable version of these nanopublications, which is very short in this case. Like in this case, we have the DOI of a specimen, which is identified as a concrete taxon name. Very short statement, but very useful. We have who is the author of this statement and the date of the publication of the nanopublication itself. So when you click on the link of the nanopublication, you will go to the Nano Dash platform, and this is the way they are presented there. So you can see the assertion part. In this case, we have the DOI of the specimen represented as a taxon concept, and the taxon concept has a name which is, in our case, identifier. We have also the provenance part, where it says that this nanopublication is derived or extracted from a particular research paper with the DOI number mentioned there, and who did this. For the concepts, and for the, in this case, for the taxonomic name concept, we are using either Catalog of Life identifiers or checklist bank. In the case you refer for a new species description, we are using zoobank identifiers because, of course, these species are not yet in the Catalog of Life or checklist bank. And for the connections between the different statements, we are using existing ontologies. The nanopublications are also very good for annotating information, and we have created different pilots in our journals. So one of the ways you can do this is basically you can comment on a research article, and if you like, you can publish your comment as a nanopublication. all nano-publications which are published on NanoDash will also be displayed alongside the article they are mentioned. And also, if this nano-publication refer to a particular part of the text, you can easily can click on it and will be navigated to the very same place. So, so I would like to say that the nano publications are self-enclosed graphs. Being RDFs, they are fair self-enclosed graphs. They can be attributed and cited. They can be generated both by humans and machines. They also can support specific templates according to the data you would like to capture. And they can be applied to any online resource. For example, the Disco infrastructures, they can be used as a mechanism for annotating information, and they really can be very powerful annotation tool and a way of sharing information. So pretty much that was from me. Keep in mind that the smallest publishable bit of information also matters. And especially when you do it in a way that easily can be captured by the machines, yes. Thank you very much.
Thank you, Theodor, for your interesting presentation. Are there any questions in the Zoom meeting? Okay. No questions in the Zoom meeting? Yeah, I see.
Is this one on? Yeah.
Yeah.
Cool.
Go ahead, please.
So one quick question. The nanopore publications are great. Are they reviewed in any way? Is there like a peer review situation for normal journals? Do they get reviewed in?
Yes and no. So you can publish nano publication directly on NanoDash, and in this case, this will not be peer reviewed. What we did, actually, you publish nano publication, you put the link in your research article, and then the research article itself going through peer review process, and the reviewers also check the nano publication, which you can update if needed.
Okay.
So yes and no.
So the expectation is that the nano publication is done before the peer review by the authors of the paper originally, or you can add them afterwards?
Yes,
as well?
in the current implementation, we are doing in this way.
Okay.
But as I said, actually the nano publications could be generated also automatically. And if we extract the nano publications from, let's say, a research article, which already passed peer review, it's also kind of, yeah.
Cool, thank you.
So I've blogged about this a little bit. There's one thing which sort of worries me about this is the value of these is obviously their connections. So you've got connections to ORCID for the authors, connections to the DOI. I'm curious as to why you connected to checklist bank. The reason for that is presumably what you want is for the text side. You want to connect to another source that's online, has RDF, and away you go to the knowledge graph. Now, Catalog of Life will give you that because they've embedded RDF. Checklist bank doesn't seem to me to do that. So it seems basically you hit a brick wall once you have identifiers that don't let you to go further through the knowledge space.
So we did, because yes, as you said, we try to first to look in Catalog of Life. But if the species is not in Catalog of Life, what we can do? If it is a new species description, of course, it is not there. So that's why we are using zoobank identifier to uniquely identify the taxon concept we are referring to. And if there is no zoobank identifier, then we add the checklist identifier, because eventually you can reconcile in the later stage this statement.
Yeah, but there are clearly identifiers that are available in Catalog of Life, and yet the nanopubs use the checklist bank. I'm kind of curious as to why, which raises the question, are they going to be curated? Like if they do actually appear linkable, is somebody going to come back and update the nanopubs in some way?
Could be, yeah. I mean, there is a mechanism for that if you like to do this. So, yeah.
Dimitri and then Lyubo.
Maybe Lyubo, you want to reply to what Rod said? Yes. You want to go first? Yeah.
Yes, just to reply to the very good question of Rod. Actually, we don't use the entire checklist bank. We use a subset of data, which GBF created especially for that purpose. And that subset includes only trusted sources of data, and these are Catalog of Life, IPNI data, Mycobank and Index Fungorum data, and Zoobank data. We don't use all identifiers used in all checklists in checklist bank. So it's a special subset of, let's say, trusted data, because Catalog of Life doesn't include all valid names. That's the only purpose. And when it becomes the next version of Catalog of Life, we can use only it.
Dimitris?
Yes, thank you. I've been struggling since that week in Costa Rica with nanopublication concept, to be honest. And the reason it's difficult, because the nature of my work is to live in two worlds, the databasing world, the Tadwig world, and the world of academic research.
Yeah.
My researcher self says no. There shouldn't be no preprints, no nanopublications, only peer-reviewed papers. My Tadwigian says, wow, this is cool. Everything should be nano and with DOIs and identifiers. So I don't know what to tell to my GBFian, and I would like to give you an example. As Lyubo knows, and many of people know, we'll go to Central Asia in November, where people don't know what Tadwig is, what Darwin Core is. I'm 99% sure that the concept of data paper will be received very well and fly very high. If we throw in the nanopublications there too early, my fear is that this will trigger salami publishing because of the concept can be misunderstood and can be used just a mechanism to inflate the DOIs to boost your Google Scholar and whatever. So I have no solutions for this, and maybe a longer answer is in place. So I just want to throw it here in the bigger room to kind of invite others to think about this matter. Thank you.
Okay, thank you, Dmitry. It's absolutely a valid concern, but as you said, we cannot prevent the way the users will use this technology. This is something we cannot do. Our understanding is that we need the right balance, and we recommend to publish only very few statements from your article which actually doesn't make sense. And in his presentation, Rod says that we're missing the link between the taxon name and the taxon name published in a paper, for example. This is something that you can very easily can express using this technology, and this easily can be captured, for example. Or the example I just showed in this presentation, that you can say this DOI of my digital specimen is identified as this concept and is published in this paper. Basically, 2 nanopublications, which tells you a lot. So that's the idea.
Okay, we need to move on with the next presentation. I would like people to remind that we have some time for discussion in the second part of the session. So please save your further questions for that, or put them in the Oxford Journal chat for that session. So let's go on to the next presentation by Joana.
Hi, thank you, Wouter. So I'm Joana. I am a biodiversity project manager at the European Bioinformatics Institute, most precisely at the European Nucleotide Archive. And I'm going to talk about the work that we did within BiCIKL and continuing about linking molecular and biodiversity data. So we all know this graph from Rod, thank you for this, about the importance of linking data, where we can see the different areas of biodiversity research and how they should be linked. And we all know the importance of fair data, increasing accessibility, findability, and interoperability. And in this presentation, I'm going to mainly talk about sequences. So sequences, major repository is the INSTC, the International Nucleotide Sequence Database Collaboration, which is composed of 3 main databases: the DNA Databank of Japan, the DBJ, NCBI in the United States, and the European Nucleotide Archive in Europe. So I'm basically going to talk about the developments that we made mainly at DNA and with the collaboration of all BiCIKL project partners regarding this data linking. So here on the left, we can see molecular data as we store them at INSTC. And on the right, we have a number of institutions and project partners representing the different areas that we want to link to, namely specimens in natural history collections. Sequence also always relate with a taxonomic identification, so taxonomy databases, literature, and also data aggregators. And within this work, we focused a lot on integrating and linking data, improving data submission workflows and tools for the uptake and implementation of data standards. We also worked on developing and improving content and enrichment workflows, so to curate legacy data that is available. And we started discussions on molecular ecology data publication. And here I'm mainly referring to environmental DNA and ASV and OTU tables publications. So regarding biodata linking at DNA, so we have a service which is called the cross-reference search, to which we link actively with several resources, some of which are EBISO resources as Magnify, Ensemble, and other resources, but we also link with external resources, including literature resources, and within BiCIKL, we increased our linking to Treatment Bank, to Open BioDiv, natural history collections that, to which we started the linking, and we should be ready soon with the GGBN, and also improving our links with GBIF and with Unite and PlutoF platforms. And how are these linking represented? So we have a source primary accession, which links with the data resource. In this case, this is an example of Treatment Bank, where we see the persistent identifier of Treatment Bank there. And then we have the target primary accession, where we have the sequence accession in this case. And we can see that the sequence record refers to the Treatment Bank accession, and then, which though it's not displayed here, the Treatment Bank also has a reference to the sequence, and you can link from there between the two resources. So a bidirectional linking. Another aspect that we tried to work a lot during this project was on improving standards in source information. So what I'm presenting here, it's not directly linked with BiCIKL, but it was a development that was made on INSTC last year, which is the improvement of the INSTC spatial temporal metadata with the minimum standards, where now country and date is mandatory for all data. And that was reflected in all of our checklists, and we can see an uptake of the number of samples that now have country information increasing, and we expect this to continue, the gap to continue to increase over time as we have better data coming in. Another important aspect is the linking with the specimen, so with the voucher, with the data from which the sequence was originated. And that's usually, so on these sequence repositories, this information is usually captured on these three qualifiers: specimen, voucher, curto collection, and biomaterial. These qualifiers have a specific format, which follows a Darwin Core standard, which is a Darwin Core triplet, which includes an institution code, a collection code, and the specimen or culture or material ID. Unfortunately, this is not something that users follow that much. Some provide very accurate information, some just provide the specimen ID, which makes it difficult then to make a proper link. So many times this information is ambiguous. So we try to build on tools that would help facilitate, one, help users provide better information upon submission, and two, to try to go into records that already exist and try to link them with the specimen of origin. And we did this through two tools that were developed. One was the ENA SAH API, Source Annotation Helper API, and the other one was a tool that was developed by BGBM, which is this SPACE tool. And we're also in discussions with BiCIKL to then implement the persistent identifiers once they become available. So DNA source attribute helper is a small API with a user interface that can help people submit. So if you know the museum where you have your specimen stored, and if you know the identifier of that specimen, then you can use this API to look for the correct code that associates with your museum. You can input the collection, and then you have the attribute in the correct format. This API uses the biocollections from NCBI because that's what's being used within the sequence data, and it goes and picks up the updated information from there. The specimen registration search engine was something that was developed by the colleagues from the Botanical Garden and Botanical Museum in Berlin. And so this is a tool that can input CSV, a text file, or a flat file that will have the sequence information and the metadata, so the attributes. And if that specimen voucher attribute is not complete, it can pick up the specimen identifier information and other attributes like name, like taxon name, date of collection, and it goes into the specimen information systems like Visco, GBIF, and Addink Bio and looks for that information. And then it outputs the best result that it finds according to that, and then the user can use that information either to correct data that is available or to in the sequence submission. So another tool to facilitate these linkings. One other aspect that we have been working on is content enrichment workflows, and this is done through a tool that has been developed previously by Elixir, which is called the Contextual Data Clearinghouse. So this allows, through the use of an INSDC accession, to submit a curation on the metadata associated. So if you know that the taxonomic identification of a sequence is wrong, you can submit that information, or if the country is wrong, you can log in and submit that information. And within BiCIKL project, the team from Tartu University, Pluto F, developed a workflow for the submission of these annotations to sequence data. This is the annotation workflow they have in operation. So they download data from INSDC. Pluto F users do their annotation. These annotations are reviewed by Pluto F team, and then they're submitted through ENA, through the Elixir clearinghouse into ENA. How we display this? So these annotations do not change the original data because that is submitted by the user, but they are displayed as third-party curations together with the original metadata. For now, we have over 30 million curations, not only coming from the biodiversity side, but also from the health data. These include taxonomic identification or other contextual metadata. We have marine projects pushing through a lot of metadata around geographical determination. And we have had over 15,000 curations that have been submitted to Pluto F through these services. So finally, we have also been discussing with experts requirements and standards that are needed for molecular ecology data representation. And as I mentioned before, this refers to environmental data and the sequence data that is derived from there. And we have ongoing development of a data model for ASV and OTU table deposition. We are also working together with other projects on these eDNA standards, and we had two talks about these projects yesterday afternoon on session 28. So beyond BiCIKL, we'll continue the work that we initiated in BiCIKL towards the biodiversity supergraph, hopefully in a BiCIKL Plus. And we'll also continue interactions with the biodiversity community and ongoing projects towards improving standards for FAIR and linked data. And these are some of the projects that we are currently collaborating and which we are working towards better data. Thank you.
Thank you, Joana. Leo, are there any questions online? Any questions from the audience? Yes, Klaus, go ahead.
Yeah, sorry, probably a bit of a high-level question, but is your entry point in the omics world always the sequence, or do you plan in BiCIKL, for example, if you want to address signaling pathways or pathogen relations, probably proteomics might be interesting here, is also the idea to include something like UniProt, for example, to align with that, or is the idea always to go from then the Elixir way, so to say, from ENA and so on, or another INSDC database to the proteomics?
Yes, so I think if I'm answering correctly, so we hold the sequence data. We do link with UniProt, and we do link with other omics services, both within EBI and outside. And I think we'll still be the sequence repository and do this linking. And hopefully in the future, we can improve these links to sequence data to all of those omics services that surround us. So that would be ideally the future, I guess.
Yes, what?
As someone who's also worked on a service to link specimens to identifiers, I know it's a difficult problem. Darwin core triplets, been tried, not really worked. What about plant specimens? Because I'm not a botanist, but my understanding is plant specimens are not typically having museum, they certainly don't follow Darwin core. It tends to be collector, name, and number. So when you go through GenBank looking for vouchers for plants, that's what you see. You do not see anything that comes close to a triple core. Does the system include plants?
Well, yeah, the idea it would be that the system would include everything. I mean, what we currently have is these Darwin triplet standards. We are discussing improving it to include these persistent identifiers that are being generated. But keeping in mind that in the beginning, not everything will come out through the persistent identifiers. We need to keep the previous standards. And within the current system, you can submit person plus identifier, though I guess this is not very resolvable in the future. I guess what we need is that systems exist for the different types of specimens that we can all put into the same system and be able to link everything, I guess. But it is complicated.
Okay, I will tell a little bit more about the digital specimen identifiers in the next talk. We need to move on. So I will continue with the last talk. This is about citing the digital specimen DOI. So let me start by explaining what a DOI is. You probably have used them already, but it's good to know a little bit more about the inner workings of a DOI. A DOI is a digital identifier of an object, which is designed to persistently identify the object. It does that through a consortium of providers of these DOIs in the DOI Foundation. It is a string in the format of a prefix/suffix, and it has a handle record with some metadata. So it's based on the global handle system, and it is resolvable through a proxy, doi.org. And the handle record is something that you not usually see, because the handle record redirects you to the thing that it identifies. But usually the handle record in the DOI only has the information about the URL it redirects to. But the handle record can provide more metadata. And in the case of a digital specimen, we actually use the handle record for more information about the digital specimen. And this allows machines to easily navigate through the space of data, look at the metadata without having to resolve to redirect to the full data to identify if the data is of interest. So this is how it works. Because these digital specimen DOIs are just DOIs, you can use tools like DataCite Commons. And here as an example, so you can just enter the DOI for specimen, and then you see some metadata about the specimen displayed. Currently, these objects are registered in DataCite. So DataCite is the register, and we work together with them as datasets. So a digital specimen is a dataset in DataCite. You can also use this citation formatter provided by Crossref. So you can enter a digital specimen DOI, and then you know how to cite the digital specimen, and you can also choose between different formats there. So all these things work out of the box just because it's a DOI. So what are the prerequisites to get digital specimen DOIs? Well, first of all, you need to have some specimen information published online. At the moment, we only support Darwin Core and ABCD datasets. So, for instance, you can have an IPT or a BioCase installation. The resource needs to be published by a CC0 or CC BY license. That means that we do not support non-commercial license, and that's because we want to fully support open data. The physical specimen needs to have an ID in the collection that's at least locally unique. That will means that is unique in the catalog in the system that we harvest from. And that is because we want to have a one-to-one relationship between the physical specimen and the digital specimen. And we do that based on the physical specimen identifier. And if that's only locally unique, we append the system identifier with that. So we have an identifier for the system as well to make it globally unique. And the host of the specimen needs to have an identifier for the institution for which we use ROR. So the digital specimen DOIs are currently registered by DISCO through our partnership with DataCite. We bear the cost for that, so there is no cost for the specimen provider involved in that. So there is a distinction to make between persistent identifiers and what I would call ordinary identifiers. Persistent identifiers are always resolvable, which means that you can put them as a URL in your browser and they will resolve to something. And a PID record has also metadata providing some context about the object that it identifies. If you want to use these identifiers in your applications, let's say that you want to create a survey, then what we often see happening is that people have some idea, okay, these identifiers, in this case an ROR for the institution, is of some importance. So they add that to their survey form. So people are asked now some extra information to ask. So it does not really help the people who need to enter the survey, because now they have to enter extra information. So it would be much better to use something like a type of head field, where you just enter your institution name as usual, and that then should automatically find the ROR plus the metadata that's assigned to the ROR, like the country in which the institution is in. Unfortunately, commercial solutions for surveys like Google Forms, Cognito Form, etc., do not support these type of head fields which need to make an API call. I don't know why. Maybe it's because of security issues. So you need to do really a custom implementation in HTML for that, unfortunately. If you are not able to do that, if you want to use these commercial tools, it may be an idea to either have a link to ROR so that people can do a lookup themselves, or just add that information later once you have received the answers of the survey. Here's a simple example of using these digital specimen DOIs in a Google spreadsheet. So if you have the digital specimen identifiers already, then you just need a little app script to fetch the metadata that's in the DOI handle record. And then you can have just something simple like import JSON from DOI, and you get the values that you are interested in. And we have an example for that, so you can just copy paste the code for that. How to find what metadata a DOI has to offer. You can easily do that by appending the DOI with no directs. And then you get, it doesn't do the redirects, and you get the handle record itself to which it resolves. You can also use the API for that, and then you get a JSON with the same information. Another example, you can also add, once you have the DOI data directly from the digital specimen, because you can find the URL to which the digital specimen redirects in the DOI handle record. It's also possible to do a lookup if you have only a local identifier. If you have the catalog number of a record identifier, you can look that up to get the DOI. We will provide some services for that. It's a bit more complex because it can be only a local identifier, and then we need some more information about in which local system it is used. Few more tricks with the DOIs. You can use the DOI also to go directly to the specimen catalog record. Not at the moment, we plan to add that to the metadata by adding an extra attribute that you are interested in the catalog view of the specimen. And you can also append the DOI with a specific version if you want to go to a specific version of the specimen. For showing specimen DOIs in a web page, we have some example code how to do that. And because we have the metadata in the DOI record, you can easily have a tooltip that provides some extra information about the DOI, like the name and the provider of the specimen and the physical specimen identifier. Now a real example, we have a publication that is just out. That is the first publication that makes use of these digital specimen identifiers to refer to the specimens. It's an interesting publication because it combines classical taxonomy with towards modern taxonomy, and the main author is 93 years old. At the same time, this publication makes extensive use of identifiers, including the nanopubs. Thanks to Pensoft, ARFA already supports these digital specimen DOIs, so you have the tooltips within ARFA and within the publication. They are also used in the publication for referring to the material citations, where the holotype in this case is described in Darwin Core. Unfortunately, there is no field yet in Darwin Core for these DOIs, so we used author catalog number at the moment, which is not the best solution, but it's the only thing that we can do at the moment. And here you have the example of the nano publication that was already shown in the previous talk. Because the digital specimen can have many entity relationships in it. So this is only one entity relationship that is expressed in the nano publication, but in a digital specimen, you could publish all these relationships as nano publications. Next steps. By the end of this year, we want to have the digital specimen architecture that we developed partly in BiCIKL available in production as a minimum valuable product. And we want to create a couple of million, about 2 million DOIs for the first specimen datasets. Then in next year, we want to launch a process for creating more digital specimen DOIs, and we will work with DataCite also on optimizing their infrastructure for that. We will work with GBIF on the publication of specimen data and digital specimen data and DOIs in GBIF. We will work with the providers on additional routes of creating digital specimen and DOIs, for instance, through digitization streets or when people are collecting in the field. And then in 2026, we aim to start a full operation of the DISCO data infrastructure. And we hope that by that time we are at nearly 100% coverage of DOIs for specimens. We aim to create specimen DOIs for all the specimens that are published in the world, not only in Europe, just as a service to the global community. Because many institutes expressed their interest of having these DOIs, but they always had concerns about the costs. Of course, we don't have unlimited budget either, so we will see on with time frame we can do that. To the end, some acknowledgements. So I want to acknowledge the DISCO development team at Naturalis, Sam Leeflang, Solein and Tom, and also our data architect Sarif, who is also here in the room. Also Jeremy Miller for our cooperation on the publication, and of course all the BiCIKL partners in the BiCIKL projects, including especially Pensoft. You can find this publication online if you follow this link. Thank you.
We got some minutes for questions. There are no questions online, so maybe from the audience. Actually, I have a question, Wouter. Yep. Is there a process in Teodor to maybe to create a specific term or field in Darwin Core for DOIs? Or?
There's nothing yet, but in the maintenance group you can make proposals for changes or for additions to Darwin Core, and we aim to make a proposal for that soon. Yeah.
Yeah, that's on. So it's sort of it's half a question, half a comment. So I'm Rory Edmonds. I actually work for DataCite. I'm leading the partnership with the IGSN, the International Generic Sample Number. So I just wanted to sort of say that I guess the one thing that was missing from your roadmap, I guess, is maybe a conversation between the IGSN organization and DISCO, because I think obviously DataCite has a strategic partnership with both, and the IGSN is on the sample itself. Here we're having a DOI, so both the DOIs, here a DOI on kind of the digital version of that sample, yeah, or specimen. So I think there's some kind of interesting things that we kind of need to figure out in the future about how this all sort of fits together in, yeah, a holistic way.
Yeah, that's a good point. So IGSN and DISCO are doing different things here. So we use DOIs for the identification of the digital objects, where IGSN does it for the identification of the physical object. So it is an alternative for your physical identifier that you use in your collection. You can use IGSN for that, but there are also many other systems in place for the physical identifiers like ARCs and DOIs and local identifiers in any form. But there are obviously some metadata that is similar, is the same, that we want to share, metadata describing the specimen that distinguish one specimen from another. So there's I think where we can cooperate.
And there's an online question from Roger Haim. Will specimen DOIs redirect to the institute that holds the specimen or only to the DISCO web page?
Yes. So if an institute, as I showed, provides us with a resolvable identifier for their catalog. So if they have a catalog online with a catalog record for the specimen, we will include that in the DOI. So you can, from the DOI, go directly to the institutional record in that case. And we also include the ROR about the hosting institution. So through the ROR, you can also go to the website of the hosting institution.
So the answer is yes.
Yes.
In case the institution has online portal with specimens. Yeah. Rod?
You're going to be sick of me at the end of this. So you have a DOI for a specimen. If I just have a specimen code, so I looked at one of these DOIs and there's some specimen in naturalis. If I'm looking at that specimen and have that code, how do I discover the corresponding DOI? How do I know if a specimen I'm looking at has a DOI?
Yeah, so for that we need to provide a custom service where we index all the specimens that have a DOI together with their physical identifier, so that you can have a lookup there. Yeah. So one issue we have with that is that we use the system identifier, and you may not know the system. So you know the institution, but in the institution it may still not be unique, because there may be several collections that use the same number 1, 2, 3. Ideally, you would like to have then the collection, an identifier for the collection, because you can assume that it is unique in the collection. Although it's also not always the case historically, but we then ask people to prepend or add a suffix to make it unique for the collection. But at the moment, there is not an agreed system for collection identifiers, and people usually do not share that in their data. So we just don't have the data. So there are some issues with that. Yeah.
Yeah, please.
Thanks, Wouter. I'm wondering how duplicate specimens in different institutions of basically the same specimen, whether some link in that, or would they all have different DOIs, or is there some way of linking this specimen in New York Botanical Garden to this one in the Cube?
Yeah, every object that has a physical identifier in a collection is for us a specimen. So it is not the organism or something like that. So it is the object that has an identifier in a collection that is in DISCO concept a specimen. So you can have duplicates for instance, of a herbarium specimen in different herbaria, but for us these are different specimens. However, you can link them together, so you can create an entity relationship that specifies, okay, these are both received from the same collection event, etc.
Marcus?
I think this needs to be the last question, because we are
probably a short one. Can you just explain why occurrence ID is not suitable for the DOI?
Occurrence ID is not suitable because we want to have identifiers for the physical objects in the collection, and an occurrence can be multiple physical objects or can be part of it. So there's a many-to-many relationship between an occurrence and a specimen in principle. Usually it's a one-to-one, but not always.
Okay, and this was the last question.
Okay, thank you. It's now time for coffee break, and then we will reconvene after the coffee break here.